Yeah, thank you. Good morning, students. Good morning, everyone. Good morning. Whether I'm audible? Yes, kindly reply whether I'm audible. Yes, good morning. Good morning. Whether I'm audible? Yes, 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 yes. We are going to discuss previous year question paper for 2023 shift 2 today. Okay. This is another important paper. So much of conceptual questions they have asked. Okay. Listen properly. We are going to explain each and everything clearly. Okay. This will be highly useful for solving your this year examination. Okay. Be patient and listen carefully in the class. Okay. Kindly take notes also. Okay. First question, what they are saying? First question, which one of the following monounsaturated fatty acid is not an omega-9 fatty acid? They are saying which is not an omega-9 fatty acid. I am going to say one trick to you now. If they have given the number of carbon atoms, if they given the number of carbon atoms, and if they given the number of double bonds, and they asked to solve which is an omega 3, omega 6, omega 9, like that means you have to do one thing. You have to write the number of carbon. For example, 18 carbon is there where the last double bond is there. For example, last double bond is present at 12th carbon means minus 12. So it is an omega 6 fatty acid. Clear? This concept is clear. So now here they are asking which is not an omega 9 fatty acid. Simple thing. 16 minus 9, 22 minus 13, 18 minus 9, 24 minus 15. So what? This is omega 7 fatty acid, clear? So palmitoleic acid is an omega 7 fatty acid and it's not an omega 9 fatty acid. Yes, this is the simplest form. Okay. Second question. The decreasing order of numerical value of molar excitation coefficient of following amino acid at 280 nanometer. We know this amino acid, particularly aromatic amino acid, will absorb light. Okay, at particular level, that is at 280 nanometer, it will absorb. Okay, they are asking the order which will have the higher molar coefficient among the aromatic amino acid, which will have the higher molar coefficient, followed by which one have the lower. Yes, option A. Okay. Okay, we can solve here. Okay. So tryptophan, you have to remember tryptophan will have higher absorption compared to tyrosine, compared to phenylalanine. Okay. But cysteine will have less one. Okay. Cysteine doesn't absorb. It will not absorb. Okay. At 280 nanometer. Okay. Why tryptophan will have more absorption? Why? This question has to come. Why? Because of the presence of that indole ring. Okay. This indole ring, okay, that will absorb more energy at 280 nanometer. Okay. Tyrosine, because of this phenol group, okay, because of this phenol group, absorption will be more. Okay, clear? So the option will be what? First absorption will be there for tryptophan, followed by tyrosine, followed by phenylalanine and cysteine. So option A will be the right option. This is another important question. This we have seen. We know the basics. Just we are going to apply the concept here. See here. Equal volume of 0.1 molar acetic acid and 0.1 molar sodium acetate are mixed. Okay, acetic acid and sodium acetate, okay, are mixed to form a buffer. Okay, consist, they have same 0.1 molar of acetic acid and 0.1 molar of sodium acetate was mixed in an equal volume to form a buffer. Now what they are asking, considering that ionization of acetic acid, they are saying ionization of acetic acid. This is acetic acid. If it is ionized means it will produce acetate ion plus H plus ion. So this is the ionization occurring at dissociation constant of, they have given the dissociation constant, that is Ka value they have given 1.74 into 10 to the power minus 5. Now they are asking what is the pKa value? What is the pKa value? TK. We know pKa formula is what? pKa is equal to negative logarithm of Ka. This is very simple one. So, negative logarithm of Ka value is 1.74 into 10 power minus 5. Okay. Just log A into B, log A plus B I am writing. Okay. Log 1.74 plus log 10 power minus 5. This if I further simplify, what it will come? Log 1.74 value is what? 0.24. 0.24 minus 5. Clear? This will come minus 5. So if I further simplify this one, we will get minus 0.24 plus 5. If we simplify 4.76, it will come. Clear? So the pKa will be 4.76. This is clear, students. So pKa will be 4.76. This is direct question. This in our class itself we have told. The pKa for acetic acid is 4.76. Okay, clear? So 4.76 will be the right option. This is simple question. I know you will solve. Just let me know. Assertion and reason they have given. The fluorescence of green fluorescent protein. Green fluorescent protein fluorescence occurs without the assistance of helper molecular prosthetic group. They are saying the green fluorescence will be happen without the help of any prosthetic group. Why it is happen means there is 3 amino acids, serine, tyrosine and glycine, okay, that will form themselves a chromophore. Okay, that will form themselves as a chromophore that imparts light transducting capability of green fluorescent protein. What will be the right option, student? What will be the right option? Green fluorescent protein. They are asking about green fluorescent protein. Yes, yes, correct. Both things are correct. Yes. Green fluorescent protein, it is a cell catalyst molecule that will not require a chromophore or prosthetic group. Okay. The amino acid, you have to know the position of this amino acid. Okay. Serine will be present at 65 position, tyrosine at 66, glycine at 67. Okay. Just you remember this also they can ask. Then they can ask from where this green fluorescent protein has been isolated. Any guesses? This we have discussed. From where? Aquaria Victoria. Yes, very good. Okay, Aquaria Victoria. Okay, from this they have isolated the green fluorescent protein. Yes, students. Next. This is very simple question. They have given different alcohol form of the sugar. We have to match with its property. Okay. Sorbitol, mannitol, myoinositol, ribitol. This we have already seen. Ribitol is an what? It's a constituent of flavin coenzyme. Yes, we have discussed in our vitamin class. Ribitol is a constituent of flavin coenzyme. It will impart yellow color. In previous year question paper we have seen it. So it will impart yellow color. So third will come option 1. Where is coming? Only here it's coming. Okay, third will come option 1. Then another important things also we can solve. Yes, very good, Budaya. Myoinositol. It is an what? It's a cyclic sugar. Cyclic sugar alcohol. Yes, this is correct. Sorbitol and mannitol. Mannitol. This we will be regularly used. Okay, most common osmotic diurent. Mannitol is a most common osmotic diurent. This is also we know. Then sorbitol. This will act as a laxative to relieve constipation. So 2413 is the right option. Yes, very good. Next, conceptual only. See, you have to observe, you have to solve properly. Okay. The molarity of buffer solution. They are saying the molarity of buffer solution made up of weak acid and its conjugate base. Okay. We know buffer means it will made up of weak acid and its conjugate base. Okay. But the molarity will be differ for both the things. That acid and base will have different molarity. Now they are asking what will be the overall molarity if we are taking 0.1 molar acetic acid, then 0.1 molar sodium acetate. What will be the final molarity of this final compound? Like that means what? It's just an additive process. It's a sum of molar concentration of both weak acid and its conjugates base. Clear? So just we have to add the molarity of the weak acid and its conjugate base. Clear? So this will be the right option. Another important question from basics only they have asked so much question. See here, which of the following pattern is true about relationship between 3 pKa values of triprotic phosphoric acid. First, you have to know what is triprotic phosphoric acid. H3PO4 is a triprotic phosphoric acid. 3 protons will be there. Okay. So this, if it is ionized, if it is ionized, it can form H plus and H2PO4 minus. Clear. So this is first ionization. Okay. If this is further ionized, it can produce HPO4 2 minus. Okay, this is pKa2. If this is further ionized, it can produce H plus and PO4 3 minus. I hope this is clear. They are asking what will be the how the Ka will change after the ionization, and they are asking what is the true relationship between these 3 pKa, like that they are asking. You see, this is acidic form. This can be predominant. Okay. So, for example, pH is 7. This is neutral pH. So, H3PO4 will have what? More acidic pH. Okay. More acidic, that will come under, for example, 2 or 2.3 it will come. Okay. So, if it donates this one, it will comes under the range of around 6.7. If it donates another one molecule of protons, this will become what? Completely, it will become an proton acceptor form. It will come behind the 7 pH. For example, 8 to 10 it will come. You see here, this is pKa1, pKa2, this is pKa3. If you see the relationship, pKa1 is always greater than pKa2 is always greater than pKa3. Clear? So, this will be the right option. Yes. Next, simple thing about glycolysis they are asking. Okay. Glycolysis. Which of the following enzymes of glycolytic pathway has achieved the catalytic perfection, in sense that an increase in the catalytic efficiency, even if we increase the catalytic efficiency, that would not increase the rate of reaction catalyzed. Okay. Even if we increase the particular enzyme reaction, among this, even if we increase particular enzyme rate, the rate of the glycolysis will not be increased. Which is that enzyme? Which is that enzyme? Whether hexokinase or pyruvate kinase or triphosphate isomerase or phosphofructokinase. What will be the answer? Yes, what will be the answer? Whether it's hexokinase, pyruvate kinase or triphosphate isomerase or PFK. We know PFK is the regulatory enzyme. PFK is the regulatory enzyme. But in question, what they are asking? Which would not increase? That's why I am saying you have to read the question properly. Which would not increase the rate of reaction? This is one of the regulatory enzyme. This is pacemaker of glycolysis. Okay. Why we are saying this is the first commitment step. Okay. If this step happen means that will enter into glycolysis. Okay. Other regulatory enzyme is what? Hexokinase. First reaction, glucose will be converted into glucose 6 phosphate. This is also irreversible reaction. Okay. Pyruvate kinase, last step for converting phosphoenol pyruvate to pyruvate. This is also irreversible reaction. So this is also one of the regulatory enzyme. But if you see, they are asking, even if we increase particular enzyme concentration, that will not increase the glycolysis. So this is the right option. Okay. They are asking which is not increase the pathway. So triphosphate isomerase, it is not a regulatory enzyme. So even if you increase the activity, it will not increase the glycolysis. Clear? So the option will be C. Non-GM herbicide tolerant rice variety PB1979, PB1985 have been developed by mutating gene coding for which one of the following enzyme? This while taking the amino acid metabolism, I have told. Okay, while taking the amino acid, I have told. Okay, they have developed a non-GM herbicide tolerant variety. Okay, by particularly mutating a amino acid biosynthetic gene. Okay, amino acid biosynthetic gene. Particularly, if you see branched chain amino acid biosynthesis will be mutated by EMS treatment. Okay, EMS treatment. What will be the right answer? Which gene, which enzyme that was regulated? acetolactone synthase, ठीक है, ALS acetolactone synthase, ठीक है, acetolactone synthase. EPSP, this is what? This is aromatic amino acid synthesis, ठीक है, aromatic amino acid synthesis pathway it will come. This will be, if this will be mutated means glyphosate resistant will form, ठीक है, glyphosate resistant will form, ठीक है, clear, clear. Acetolactone synthase will be the right option for that, ठीक है. Next, given below are two statement. One is assertion and another is labeled as reason, ठीक है. This is very simple question, ठीक है. Carbohydrates are most efficient storage form of energy as compared to triacylglycerol. Whether it is true? No. Triacylglycerol are more efficient form. Why? Because this triacylglycerol will not require water molecule. This is hydrophobic molecule. So the body doesn't need to carry extra amount of weight for the water. Okay, so there that no need to be hydrated. And second most important thing, the carbon atoms that are present in fatty acid will be in more reduced form. So if we oxidize this one, more amount of energy will be produced. That's why triacylglycerol will be act as a major storage form of energy in mammals. Okay. So you see here they have given carbohydrates, so this will be wrong. And they are saying the reason is carbohydrates are more oxidized and hence yield more amount. This is also wrong. Okay. If a compound is reduced one, that if we oxidize, that will give more amount of energy. Okay. So the both statement are wrong. Both A and R are false. Clear? They have given here different forms of DNA, A DNA, B DNA, Z DNA, A, B and Z DNA and H DNA. Okay. First, the most important thing is Z DNA you have to think. Okay. Z DNA, this is what? It will have alternating purine and pyrimidine. This in a class I told. Purine and pyrimidine will be there. Nucleotides will be there. Another most important thing, this will be present highly in where? High salt concentration. High salt concentration. Okay. Wherever high salt environment is there, this ZDNA will be predominant. Okay. In cellular condition, bDNA will be predominant. Okay. bDNA will be predominant. aDNA will be predominant in where? That will be predominant in dehydrated conditions. Okay. Now you match bDNA, where it will come? It will be present more. This will be the most common form of DNA found in vivo. Okay. This is clear. So B will come option 2. Where is coming B? So, sorry, A will come option 2. All the things is coming option 2 only. Then aDNA, where it will come the aDNA? aDNA will come in where? aDNA will be present in desiccated condition. That's what I told. So B will come option 1. So B will come option 1. ZDNA, I already told it will be present in high salt condition. So C4, C4, okay. D, what it will come? HDNA, it is a triple helical structure found by polyurine and polypyrimidine structure of DNA. Okay, that is a triple helical structure. So Z will come 3. Okay, clear? 2, 4, the both they have given same option only. Okay, clear? Next, see here, repeatedly they are asking this question from spectrophotometer. Okay. In 1 year, they ask the formula. Okay. In 2025, they ask the formula. Okay. Formula is what? Absorbance equal to sorry, 2 minus log percent T. Okay. In 2024, they asked a simple problem of, like based on this formula. Okay. In 2023 also they are giving one more question. Okay. So this is the major question area in technique in biochemistry. You have to solve this properly. Okay. The absorbance of a solution of analyte having 70% transmittance. They have given percent transmittance is 75%. Okay. Would be equal to, they are asking what will be the absorbance. Okay. We know absorbance equal to 2 minus log percent transmittance. Okay, they have given the percent transmittance value that is 75. Okay, but they didn't give the direct value of log 75. Okay, log 75 value they didn't give, but they have given in indirect way 5 and 3. You have to multiply. Okay, in which multiplication it will come 75. We can write 5 square into 2, 25 into 3 is what? 75. Okay, I am just simplifying. I am writing like this: 2 minus log 5 square into 3. Clear? This I am solving only for this thing. Okay, I am solving for log 5 square. Log 5 square into 3. I am solving only for this thing now. Okay, what it will come? 2 log 5 plus log 3. This is clear. 2 log 5 plus log 3. Yes, correct answer everyone told, but we have to solve now. See. So, 2 into log 5 value is what? 0.666990 plus log 3 value is 0.4771. If you multiply this 1.875, it will come TK. So, 2 minus 1.875 TK. So, the right option is 0.125. Very good students, you have solved already. Okay. So 0.125. Okay. This is very important sum you have to solve. Okay. Next question. Van Demeter equation. Okay. Then another thing what I want to say means, see, no one can write 120 out of 120 questions. Okay. Some of the concept will be very difficult. Okay. Even if you read the book from top to bottom, every year they will ask some of the new questions, some of the terms, new cycles, everything they can ask. If you know that, if you know clearly that one, if you have any guesses, you write that one. Then if you don't know, kindly leave that question. Okay, this is my suggestion. As a mentor, this is my suggestion. Don't write much negative marks. Don't write and take much negative marks. You know, negative markings also plays a major role for the top rank. Okay. So, see, some of the terms are very difficult. Okay. That thing you kindly leave that one. Okay. Don't go and do the mistakes. Okay. Van Demeter equation describes the element of band broadening and chromatographic column efficiency. Okay. For chromatographic column efficiency, this is an equation. Okay. Van Demeter equation. This is that equation. Okay. Whether this is clear? Height equivalent to theoretical plate. HCTP means height equivalent to theoretical plates is equal to A plus B by U plus Cu. Okay. Here the component what it is saying means A means eddy's diffusion. These all are different terms. Eddy's diffusion. B is longitudinal molecular diffusion. Okay. These all are concepts. Okay. C is mass transfer between mobile phase and stationary phase. Clear? So what is this user? This is what means this is elution time or flow rate. Okay. So this will be determined. So these are the components of this Van Demeter equation. Based on this, they have asked the question. Okay. Now we have to see eddy diffusion of sunlight in a column. Yes, this will come. Longitudinal diffusion of sunlight in a column. Yes, this will also will come. They have given in the formula. Mass transfer of sunlight between stationary and mobile phase. This will also will come. Yes. Then flow rate also will come. Okay. Which biochemistry? See. So the right option for this is A, B, C, and D. Okay. A, B, C, and D. Someone has asked which will be the best biochemistry book to prefer. Okay. My suggestion is if you have enough time, you have to go through with Leninger. Particularly 6th edition is very much good. Okay. Apart from that, if you want to read more concept, you can read Techniques in Biochemistry by Pranav Kumar. Okay. Technique in Biochemistry by Pranav Kumar. I am writing the book name here. Okay. Biophysics and Molecular Biology. Biophysics and Molecular Biology by Pranav Kumar. Okay. This is outstanding book for reading technique in biochemistry. Okay. Technique in biochemistry. And for normal biochemistry, you go for Leninger. Okay. Leninger biochemistry, particularly 6th edition is excellent. Okay. If you have less time, sir, I just want to read, I just want to give a quick revision. You can read instant notes on biochemistry by Hams. Okay. By D. Hams. Okay. This is another very good book Here they have just concise the lineager and they have given the concept here. Okay. So these are the major books for biochemistry. Okay. Next question. Here they have given the different photo like C2 cycle enzymes they have given, and they are asking the location of the enzyme. Where will be the enzymes will be localized. Okay. You see first glycolate oxidase. This glycolate oxidase will produce hydrogen peroxide. So catalase should be there to degrade this one to decay, to degrade this one. So particularly this glycolate oxidase will be present in peroxisome. Yes. So A2, where coming A is 2. Okay, A is 2 is coming in this 2 option. Glycine decarboxylase. Yes, glycine decarboxylase. This reaction we know. The photorespiration will occurs in CPM, chloroplast, peroxisome and mitochondria. This glycine decarboxylase enzyme will be present in mitochondria that will convert two molecule of glycine into serine. Okay. So this will be present particularly in mitochondria. So B1, where coming B1. Okay. Glycerate kinase. Glycerate kinase, where it will be there? It will be present in chloroplast only. That will be the last step of this photorespiration. So chloroplast C4. So this will be the right answer. NADPH dependent hydroxypyruvate reductase. This is present exclusively in cytoplasm. Yes, very good, Udaya Lakshmi. Yes, very good. Now we are going to see the basic things only. They have focused much on the basic things. Okay. See here, they are asking about secondary structure. Yes, they are asking about secondary structure of protein. Okay. They have given antiparallel and parallel beta sheets. They are asking question about antiparallel and parallel beta sheets. The secondary structure of protein, parallel beta sheets are less stable than antiparallel beta sheets. This is wrong. Correct. Antiparallel beta sheets are more stable than parallel beta sheets. Okay. Why this anti-parallel are more stable? More stable means the hydrogen bonding between the sheets, between the beta sheets will be what? They will be lies on each other. Okay. You see in this figure. Okay. It will form a straight hydrogen bond in anti-parallel. Okay. Because of this, the interaction will be more between the two strands. It is the more stable one. But if we see in parallel, the hydrogen bonds will be distorted. You see here distorted hydrogen bonding will be there in parallel beta sheets. Okay, that's why anti-parallel beta sheets are more stable comparing to parallel beta sheets. In our question, they have given assertion parallel is more stable. So this will be wrong. Then another thing they are saying the hydrogen bond of parallel beta sheets are distorted in comparison to the anti-parallel beta sheets. This is true only. This is true only. Yes. So this will be right. Okay. So A is false and B is true. Clear, students? Clear. This one is clear. Yes. Next, the sequence of action of enzyme of fatty acid beta oxidation pathway. This is repeated question they have asked in 2024 also. Okay. The sequence of actions of enzyme for fatty acid beta oxidation. Solve one, let me know. Solve one, let me know. Okay. Here they have given parallel or less stable. This will be right only, correct? Parallel or less stable only. Yes, very good. Parallel or less stable only. Okay, so A is true and R is correct. R is false. Clear now? Udaya Lakshmi, whether it is right now? Just let me know. Question. Yes, very good, very good, very good. Now you have to solve this one. Solve one, let me know the answer. The sequence of fatty acid, yeah, sequence of fatty acid oxidation they have given now. You have to solve and let me know. What will be the answer? The sequence of fatty acid oxidation. First, which enzyme will come? First, dehydrogenase enzyme will come. Next, hydradase enzyme will come. Next, also dehydrogenase enzyme will come. Okay. First, acyl CoA dehydrogenase enzyme will come. Second, enoyl CoA hydradase enzyme will come. Then beta hydroxy acyl CoA dehydrogenase. Then thiolase will come at last. Okay. So DCAB. DCAB. So option C will be the Right answer. Yes, very good. When two phosphatidylglycerol moiety join with each other with elimination of one glycerol molecule, they are saying phosphatidylglycerol molecule, okay, is binding with another phosphatidylglycerol. Here one glycerol molecule is removing. So what will be the compound formed? What will be the compound formed? What will be the compound? Whether cardiolipin, cephalin, ceramide or ganglioside, what will be formed? This we have told, we have discussed in our basic class. Biomolecules, fatty acid metabolism, there itself I have told. This is another important question. This compound will be present primarily in mitochondrial membrane. What will be the right answer? Yes, students, what will be the right answer? Cardiolipin will form. Cardiolipin will form. This we can also say diphosphatidylglycerol. Diphosphatidylglycerol. Okay, diphosphatidylglycerol. Then what is meant by lecithin, ceramide, ganglioside? Okay, you have to remember another name of phospholipids also. Okay, yes, another name of phospholipids also you have to remember. Phosphatidylcholine. Phosphatidyl ethanol amine. Okay, this is ethanol amine. Okay, you have to remember what is the other name of phosphatidylcholine and phosphatidylethanol amine. Okay, so phosphatidylcholine. Okay, phosphatidylcholine is known as lecithin. Okay, lecithin. Phosphatidylethanol amine is known as choline. Okay, wherever C is coming here, the name will not start with C. Okay, phosphatidylcholine is lecithin. Phosphatidylethanolamine is choline. Okay, so here they have given cephalin. Okay, lecithin and sorry, this is cephalin. Okay, so phosphatidylethanolamine is cephalin. So this is phosphatidylethanolamine. Clear? Ethanolamine. Ganglioside means what? It will have a negative sugar molecule, that is sialic acid. Sialic acid will be there. Ceramide is what? Principal backbone for sphingolipid biosynthesis. Sphingolipid biosynthesis. This we have already discussed in our class. Just I am giving one revision. Okay. Fructans. Fructans are polymers of fructose. Okay. Fructans are polymers of fructose built upon which of the following starter unit. Okay. They are saying fructans is synthesized from which of the starter unit, which will be act as a starter, so that further fructose molecule will come and join here. What will be the starter? Any guesses? What will be the starter? Sucrose or galactose or fructose or trehalose? Yes, sucrose will be the starter. Okay, you see this figure. First, sucrose molecule will be there. Okay, sucrose molecule is there. 6 sucrose fructosyl transferase. That means from another molecule of sucrose, fructose molecule is transferring here. Because of that, the fructan was formed. That is 6 kestose was formed. Okay, you have to just remember sucrose is the basic unit. Then another molecule of sucrose will come. This will have glucose and fructose. These fructans will be transferred to the first molecule of sucrose. That will lead to synthesis of fructans. There are different classes of fructans are there. Take a leaven type, grass type, graminase type, neo type, like that is there. That is not that much important. You have to know the basics. Okay. So sucrose will be the starting molecule. Okay. Sucrose will be the starting molecule. Then another thing I want to say, raffinose class of oligosaccharide. Raffinose class of oligosaccharide. ठीक है, RFO. ठीक है, raffinose. Raffinose is a what? It's a trisaccharide. We know raffinose, stachyose, verbascose. ठीक है, this is trisaccharide, tetrasaccharide, pentasaccharide. ठीक है, these are known as, these are known as raffinose class of oligosaccharide. Here also the starting molecule will be sucrose. ठीक है, the starting molecule will be sucrose. ठीक है, the another most important thing is this will be linked with galactose molecule. So the third sugar will be galactose. Clear? So if sucrose plus 2 molecule of galactose is there, means that is stachyose. Sucrose plus 3 molecule of galactose is there, means that is verbascose. Okay, this can also they can ask. Raffinose class of oligosaccharide. Okay. Here also the backbone is sucrose only. So the glucose will come and bind here, but in fructans, fructose will come and bind there. Okay, clear? Which of the following enzyme contains an unusual amino acid selenocysteine at its active site? Which amino acid will have a selenocysteine at its active site? Yes, this is simple question. Glutathione peroxidase will have selenocysteine in its active site. Okay. Selenocysteine will be there in that active site. Now we are going to see which is the codon that will code this selenocysteine. Okay. UGA is the codon that will code this selenocysteine. Then the single letter codon for this is what? U. This is very important. These two are very important. Another most important thing is, we know this amino acid is there. Okay, amino acid mRNA is there. This will be translated to proteins. Okay. If you see in enzymes or some other things, the metal ions will be accumulated at as a post-translation modification process, correct? So the metal ions can accumulate. But the thing is, in selenocysteine, wherever in glutathione peroxidase, wherever the protein synthesis is happening, during that synthesis process itself, this selenocysteine will be accumulated there. Okay, so selenocysteine, selenium ion will be there, that will be accumulated in translation process itself. Okay, these are the important points. Okay. Here they have given the different enzymes and what are the cofactors required. Okay, enzymology question. Nitrogenase, what will be the cofactor? Yes, homocitrate will be there as a cofactor. Okay, so A will come to nitrite reductase. Specifically, nitrite reductase will have seroheme. Okay, nitrite reductase will have seroheme. This we have seen in our nitrogen metabolism class. Serine hydroxymethyl transferase. It's a PLP requiring enzyme. Okay. Transaminase, that also will require PLP as a cofactor. I am writing here. Transaminase. It is a PLP requiring cofactor enzyme. Okay. Then glycogen phosphorylase. This also requires PLP as a cofactor. Okay. The carboxylation reaction primarily requires biotin. So 4. 2, 1, 3, 4. Already answer came. 2, 1, 3, 4. Very good. 2, 1, 3, 4. So option A will be the right option. Yes, very good. Next. Given below are two statements. So assertion and reason they are giving. Citric acid cycle is a ampibolic pathway in nature. That ampibolic in nature rather than only catabolic. Okay. They are saying citric acid cycle is an ampibolic pathway because there is both catabolism and anabolism is there, like that they are saying. Reason what they are saying, several biosynthesis pathway utilize TCA cycle intermediates, precursor for synthesis of important molecules. What will be the answer? This I only took the class. What will be the answer? Any guesses? What will be the answer? TCA cycle is, yes, it's an ampibolic pathway because some of the intermediates, for example, succinyl-CoA, yes, option A. For example, succinyl-CoA that can be siphoned off for other products, other product formation like heme biosynthesis, oxaloacetate that will be involved in other pathway formations. Okay. So both A and R are correct and R is the correct explanation for A. Okay. This is another simple question from electron transport chain. Okay. The correct sequence of flow of electron between the following component of mitochondrial electron transport chain. They are saying what is complex 1, 2, 3 and 4 where the electrons will flow. Okay. First complex 1 will be there. What? Yes, we know NADH dehydration is. Then from there it will give to ubiquinone. Then cytochrome C will come. Okay. No, next BC1 will come. Cytochrome BC1 complex will come. From here, cytochrome C will take the electron. This electron will be given to cytochrome A3. Clear? Cytochrome A3. But here what they have given? So BC1 complex where it will come? B, then D. ACDB. ACDB, where it's coming? ACDB. Clear? War. So option B. Clear? Next, assertion and reason, they are asking about inhibitors of mitochondrial electron transport chain. Inhibitors of mitochondrial electron transport chain. Okay. In early part of the 20th century, 2,4-dinitrophenol was prescribed as a diet pill for weight loss. Okay. 2,4-dinitrophenol as prescribed as a diet pill for weight loss. Okay. The reason why they are saying means DNP reduce metabolic rate by uncoupling oxidative phosphorylation. So, it will uncouple the oxidative phosphorylation from electron transport chain. What will be the right answer? Yes, what will be the right answer for this? Yes, we know DNP is an uncoupler. Yes, we know that one. It's an uncoupler. Why we are saying that is an uncoupler? Yes. Option B. Any guesses? Yes, very good. A and R are correct and R is the correct explanation of A. This is very simple question only. Then they are asking about nitrogenase enzyme. This we have already discussed. Nitrogenase enzyme is considered to be a sluggish and inefficient enzyme. In class itself, I told it's an sluggish enzyme and it's an inefficient enzyme. Why? For each electron transfer, the both component has to be separated and it has to be assembled back. Okay, so it is a sluggish enzyme. Okay, the nitrogenase enzyme must go through several catalytic reduction cycle wherein the two component of nitrogenase, that is iron protein and molybdenum iron protein, get dissociated from each other following the electron transfer before final product forms. Yes, correct. Both A and R are the correct and R is the correct explanation of A. Very good. Here they have given, see, from nitrogen metabolism, another important question they have given. Okay. In our class itself, I told nitrite reductase, NIR, sulfite reductase. This both will require 6 electrons. Okay, 6 electron for converting nitrate to nitrite, nitrite to ammonia. Okay, sulfate to sulfite. Okay, this is a 6 electron carriers. Okay, you just see here where it's coming, reduction of sulfate to sulfite. This is what 6 electron carriers. So D will come option 1. Where is coming? D is coming option 1. Okay. Then another important thing, conversion of nitrogen into one molecule of ammonia. Nitrogen is converted to one molecule of ammonia. Here what? 4 molecule of NADH will be utilized. So 4 into 2, 8 electrons will be utilized there. Okay. So 8 electron will be utilized there. Okay. Reduction of nitrate into nitrite. There only 2 electrons will be required. Reduction of nitrate into hydrogen. This is specific one. 7 electrons are required. Okay, clear? 7 electrons are required. Another simple question. In 5' to 3' sequence of following loops present in cloverleaf structure of tRNA molecule will be. They are asking, if we read from 5' to 3', what are the arms that is present in the tRNA cloverleaf structure? So another thing, secondary structure of tRNA is cloverleaf, correct? Cloverleaf. What is the tertiary structure of tRNA? Any guesses? What will be the tertiary structure of tRNA? First you comment this one, then I will, yeah, BACD, okay. BACD, this is okay. First you comment this one. What will be the tertiary structure of tRNA? Secondary structure is cloverleaf structure. Tertiary structure is what? Tertiary structure will be twisted L. Okay, this is another important question. It is twisted L shape. Okay, twisted L shape. Okay. Now, if you see here, first will come D arm, then anticodon arm, variable arm, T size C arm. Okay. D arm will come. So, first will come D arm. Where D arm? D loop. So, first B will come. Okay. Next A will come. Next C will come. Next D will come. B, A, C, D. This is clear. Okay. Another important things we are going to discuss about tRNA. Okay. tRNA is the smallest RNA molecule. Okay. tRNA is the smallest RNA molecule. Okay. Then most important, I am saying comparing to mRNA and ribosomal RNA, tRNA is the smallest one. If they given siRNA or miRNA means this will be the smallest one. Okay. This is clear. Then another important thing, what is you have to focus for exam point of view is at 3' site of this tRNA, CCA sequence will be there. CCA will be there. Okay. This will be added after the synthesis of this tRNA. Why means this A will bind with the amino acid. Okay. This A only will bind with the amino acid. Okay. See here at 3' the adenine will bind this amino acid. The anticodon arm will go and bind with the mRNA, then the particular amino acid will be attached there. Okay, clear? So these are some of the important points. Another point they can ask means, so much of modified bases will be there. Okay, we know thymine will never present in RNA molecule. See here, thymine is there here. Okay. Another thing, pseudouridine is there. This symbol is for pseudouridine. Okay. What is meant by pseudouridine? Pseudouridine. Okay. The simple thing is, we know N1 will linked with carbon 5. Okay. This will be general linkage of N glycosidic bond. DNA and sugar linkage will be there. But in pseudouridine, C5 will linked with C5. Okay. This is the difference. Okay, this they can ask. Okay, C5 will be linked with C5. This is the important points you have to focus for exam. Okay. Another important question they have asked from enzymology. This is greater than. Okay, greater than. Not less than. Anyone can solve this one. This is very simple only. Among this 4 option, we know if substrate concentration is equal to Km, that is what? The enzyme reaction will be of the maximum velocity. Okay. So you see here rate of the reaction is equal to half the maximum velocity. So 3 will come option 4. Where is coming? 3 will come option 4. Simple. Okay. Work smartly. So C is coming option 4. Okay. Remaining things we will also see. Okay. If substrate concentration is less than Km, you see here this is the Michaelis-Menten equation. If the concentration of substrate is less than Km, this will be directly depend. So this is what? The enzyme rate of the reaction will be directly depend on the substrate concentration. So it will come as a first order kinetic reaction. Yes, reaction will obey first order kinetic. So it came. Okay. If substrate concentration is behind the Km or greater than Km, this will follow zero order kinetics. Okay. This is also clear. If substrate concentration is less than Km, what it will do? It will Km and Vmax do not define the enzyme catalyst reaction. Yes, very good. Already answer came 2143. Yes. Next, another question on enzymology only. They are asking in different way. Okay. Which of the following statement is true about type 4 isoenzyme of hexokinase? We know isoenzymes means what? These are different form of the same enzyme catalyze the same reaction. Okay. But this may be differ in amino acid sequence, catalytic efficiency or other kinetic parameters. Clear? So this is the simple definition for isoenzymes. Okay. Now they are saying about type 4 isoenzyme of hexokinase. This is what? This is glucokinase. The type 4 isoenzyme of hexokinase is glucokinase. Okay. This glucokinase is predominantly present in liver tissues. The major role is it has to convert glucose into glycogen molecule. Okay. Glucose into glycogen molecule. Okay. For conversion of glucose into glycogen molecule, this will be involved. This what it will catalyze means it will convert glucose to glucose 6 phosphate. You just imagine in our blood so much of glucose is there means first thing insulin will be produced. This insulin will enhance the activity of this glucokinase or it will signals the synthesis of glucokinase enzyme. Okay. So the major important thing about this glucokinase is it will not inhibit by its product. Okay. So glucose 6 phosphate is there now. This will not inhibit the reaction. Okay. First thing. Second thing is the Km for this enzyme will be more. Km will be more means what? Large number of substrate can be converted into product. For that it will take so much of time. Okay. Km means Less means affinity of the enzyme will be more. Clear? So you see here, based on this concept, we are going to solve. It is highly specific for glucose, and this often called as glucokinase. Yes, this is true. It has much higher Km value for glucose. This is also true. It's allosterically inhibited by glucose 6 phosphate. I told this will not inhibit by glucose 6 phosphate. Okay, so this is wrong. Wherever C is coming, that is wrong. Okay, it is non-inducible enzyme. No, it is an inducible enzyme by insulin. Okay, then it will also and follows Michaelis-Menten. No, it will not follow Michaelis-Menten equation. It will have a sigmoidal behavior. So the right option is what? A and B only. Yes, clear. Next, which of the following is true about the role of malonyl-CoA in fatty acid metabolism? They are saying malonyl-CoA role in fatty acid metabolism. This is very simple one only. It is an inhibitor of enzyme converted cell transferase. Yes, we have seen this malonyl-CoA will go and bind with the CPI enzyme. So it will act as an inhibitor. Okay, inhibitor of CP1 enzyme. High level of malonyl-CoA suppress fatty acid entry into the mitochondria. Yes, this will go on to bind here. So beta oxidation will be inhibited. Clear. High level of malonyl-CoA lead to reduced flux of fatty acid towards triacylglycerol biosynthesis. See here, if high level of malonyl-CoA is there, means that will signal so much of fatty acid will be produced. So TAG biosynthesis can be increased. They are saying this will decrease. So C will not come. Wherever C is coming, that is wrong only. Okay. Low level of malonyl-CoA favors fatty acid oxidation. Yes. If malonyl-CoA is low, it will not go and bind here. So CP1 enzyme will be active. It will take the fatty acid to mitochondrial matrix. So beta oxidation can be proceed. Yes. So the right option is A, B and D only. Okay. Another important question. Which of the following statement are true about bundle seed cell of C4 plants? We know in C4 plants, initial CO2 will come into mesophyll cell, that will go to bundle seed cell. Okay, here oxaloacetate will form, that will convert into malate. This will go here, here decarboxylation will happen, that will lead to pyruvate. Okay, this CO2 will be utilized for Calvin cycle. Okay, this question they will ask. Okay, in C4 plants, listen the question properly. In C4 plants, where C3 cycle will happen, whether in mesophyll cells or bundle sheet cell. So bundle sheet cell will be the right option. In our question, what they are asking, in comparing to mesophyll cells, they contain large number of A granule chloroplast. Yes, this bundle sheath or A granule. This is correct. They have very thin cell wall. No, it will have very thick cell wall. So this will be wrong. They have no intracellular spaces between them. Yes, intracellular spaces will not be there in bundle sheath cell. This is also correct only. So wherever B is coming, that will not come. Next, they generate no oxygen because of low activity of PS2. This is also correct. So the right option is A, C and B. Hereafter, very simple questions will be only there. Listen properly, okay? Which of the following phosphate is removed from the incoming nucleotide during the bacterial transcription? We know, okay, during transcription, which phosphate will be removed? For example, ATP is there, triphosphate, phosphate, 3-phosphate will be there, adenosine residue will be there. Okay, this is ATP. Okay, if some of the chain of nucleotides is here, it will come and bring a nucleophilic attack here. Because of this, which phosphate will be removed from this upcoming nucleotide, like that they are asking. It will remove both, this is alpha, beta, gamma. It will remove beta and gamma phosphate as PPI. Okay. As pyrophosphate molecule, it will remove. In our option, what they have given, alpha, gamma, 5 prime, 3 prime. Gamma is the only option given, but gamma, then beta, this both phosphate can be removed as PPI from the upcoming nucleotide. Okay. Which of the following is not related with termination of transcription in E. coli? Okay. If transcription has to be terminated in E. coli, which of the signaling molecule is not related, like that they are asking. Okay. First, we have to know what is this PP, GPP, triple P, G, double P, alarmones and PGP. Okay, this is what tetraguanosine. Okay, tetraguanosine. This is pentaguanosine. Okay, these classes are known as alarmones. Okay, that means what? If the E. coli cell have low energy status and amino acid is less in that cell. Okay, for example, amino acid synthesis is less in that cell. Now the cell will produce signaling molecules that is known as alarmones. Okay, this PPG and PPGPP is a class of alarmones. Okay, these are class of alarmones. What it will do means it will inhibit the synthesis of ribosomal RNA and tRNA. Clear? So if this both ribosomal RNA and tRNA synthesis is not there, it will inhibit the transcription. Clear? So the right option is which is not related. So what will be the answer? PGP, dinucleotide guanine will not be come. Okay. Next, about bDNA, they are asking here which of the following is true about bDNA. It is right-handed. Yes, it is right-handed only. ZDNA will be left-handed. Okay. Yes, this is clear. 10.5 base per turn of helix. This is also clear. Yes, it will have 10.5 base pair. Its helix diameter is broadest among A and Z type. They are saying the diameter of the helix will be broader than A and Z type. If you see the diameter, bDNA will have 20 Armstrong, aDNA will have 23 Armstrong, and zDNA will be slender, that will be around 18 Armstrong. So they are saying this will be the broadest, but no, this is not broadest. Okay, so this will not come. The glycosidic bond is of anti-type. This is true only. This will be there as anti only. So A, B, and D. Where is coming A, B, and D? Option D will be the right option. Okay. Then what the point you have to remember in this differentiation means you have to know this glycosidic bond will be syn or antiposition. Syn or antiposition is that DNA. Okay. Syn in syn form will be there or anti form will be there. Pyrimidines will be always present in anti form in zDNA. Okay. Purines will be present in syn form. This also they can ask. Okay. So from DNA basics, they have asked two questions. First question they have asked where this zDNA, bDNA, aDNA will be present predominant in endogenous. Okay. Whether it will be aDNA will be present in cell or zDNA will be present in high salt concentration. Like that, they have asked one question. Okay. Second question they asked about the difference. Okay. About the sizes they have asked. Okay. If you want to practice topic wise mock test, you can kindly enroll in this mock test bundle. Okay. Then you practice well. Okay. Those who are targeting for next year ICAR 2027, they can enroll in our plant biotechnology course. It will be highly useful for your preparation. Okay, students. Now, which of the following cellular structure found in all prokaryotics? They are saying in all the prokaryotics, this cellular structure will be present. Yes, flagellum will not be there, that will be present in bacterium. Okay, capsules will not be there. So, ribosome only will be present in all the organisms. Okay, prokaryotic organisms. Okay, which of the following cellular structure is found only in plant cell? Continuously 3 questions they have asked about cells only. Okay, cells and cell components only they have asked. Which of the following cellular structure is found only in plant cell? Plant cell will, yes, excellent. Glyoxysomes are peroxisome also that will be present specifically in plant cells. This will be present specifically in animal cells. Okay, clear? Yes. Which of the following can add the terminal 5' CCA at the end of the mature tRNA? This I already told. Yes, this I already told. Which will be the added a terminal 5' CCA at the end of the mature tRNA? What will be added? I told tRNA nucleotide transferase. After synthesizing of the tRNA, this will be added there. After synthesizing of the tRNA, this CCA component will be added there. Then what about this RNase P, D, E and F? This will be involved in processing. Okay, this will be involved in processing. Okay, processing of the tRNA will be involved. For example, RNase P will process this 5' end RNase E. Apart from this, RNase D that will process the 3' end. Okay, this they can ask. Okay, RNase P, RNase D, RNase E, and RNase F. Okay, which of the following cellular structure is found only in plant cell? Someone has answered option B. What will be the answer? Whether it's option B or option C, A, and D. What will be the answer? Glycosomes is specific for plants, no? Yes. What are twintrons? Twintrons. What is mean by twintrons? Okay, this is another. This they have asked in 2025 question in match the following. They have asked. Okay, twintrons means what? That are the introns present between different groups of introns. Okay, so composite structure made up of two or more introns, group 2 and group 3 introns. You have to know the basics of introns also. What is group 1, group 2, spliceosome and archaeal introns? Just you go through this one. See, group 1 intron means there will be an external nucleotide. Most important thing, group 1 and group 2 are what? They are self-spliced introns. Self-spliced. There is no need of spliceosomal complex. There is no need of spliceosomal complex to remove the introns. Okay. Group 3 is just, group 3 will comes under the class of spliceosomal mediated intron removal process. Okay. Here different spliceosomal process will happen. Okay. This you have to know. Okay. Which of the following restriction enzyme requires only magnesium as a cofactor for cleavage? They are saying only magnesium will be used as a cofactor for cleavage. We know there are three types of restriction enzyme, 1, 2, and 3. This second class is the predominant. It will use only magnesium as the cofactor. Third class will use magnesium plus ATP. This first class will use ATP, magnesium, and yes, adenosyl methionine. Okay, Sam, what will be the right answer? So, if you see the example, whatever they have given, that also I will write here. ECO R1 is the type 2 restriction enzyme. ECO KB, this you should never remember, never forget. KB, this will come under class 1, and ECO P1 will come under type 3. So the right option that is using only magnesium is ECO R1. Yes, very good. ECO R1. Which of the following is the activity of alkaline phosphatase? What will be the activity of alkaline phosphatase? Yes, phosphatase will remove the phosphate group. So removal of phosphate from 5' group end. Yes, it's very clear. Removal of phosphate from 5' end. Yes, which of the following can be used for obtaining single strand copy of a cloned sequence? This during our vector class itself I told. M13 phase will be used primarily for DNA sequencing. Because here single stranded DNA can be obtained. Yes, so yes, very good. So M13 phase will be widely used for this technique. Okay, very good, students. Next, which of the following is an example of scoreable marker? Scoreable, we can say visual interpretation we can do. Scoreable. Okay, you see just which is odd one out. Ampicillin is a what? Ampicillin, kanamycin, neomycin. This all are what? This is screenable. Screenable marker. Okay, so beta galactonase is an scoreable marker. Okay, yes, beta galactonase. Other example we can say GUS, green fluorescent protein. These are also some of the examples. Okay, luciferin. These are some of the examples. Okay. Apart from that, what will these are the some of the examples. So markers, from markers, sure they will ask one question. What are the example of scoreable markers, screenable marker, reporter genes? Okay. From that you will pakka get one question. Okay. Which of the following cannot be used for confirmation of positive clones? See, whatever we have taught you in the class, that are they are asking directly here. Okay. Which of the following cannot be used for confirmation of positive clones. Colony hybridization, yes, we can use. Colony PCR also we can use. Growing transformation and selection media, this also we can use. Antibiotic selections. Okay. So growing transformers under low temperature, this is not a selection criteria, so this will not come. Yes. Probes are used only in, this also we have seen, southern blotting, northern blotting, western or protein blotting, this we have seen. Okay. Probes will be particularly used in southern blotting, we can use. Yes. Northern blotting also we can use. Colony hybridization, here also we can use the probe. In RT-PCR, we will not use probes. Okay. So the right option will be what? RT-PCR. Southern blotting will be done for DNA, this for RNA, northern blotting for protein. This is also some of the key question they can ask. Yes, RT-PCR. Yes, correct answer, Uday Lakshmi. If they have given QRT-PCR, here we will use probes. Okay. In QRT, in quantitative real-time PCR, we will use probes. Okay. What probes we will use? Tachman probes. Tachman probes we will use. Okay. But in RT-PCR, we will not use probes. Okay. Clear? Yes. Next. A progeny Drosophila with grey body and vestigial wings derived from a cross between parents with grey body and normal wings. See, a progeny Drosophila with progeny is grey body, vestigial wings. Okay. Derived from a cross between parent with grey body and normal wings. Normal wings with black body and vestigial wings. Okay. But we are obtaining a progeny of grey body and vestigial wings. What will be the right answer? Whether there will be a recombination between alleles of body color and types of wings of Drosophila will be there, or whether there will be linked genes, whether this is a pleiotropic effect, or whether these two genes are asserted independently. What will be the right answer? Yes, this is correct. This is a recombination between alleles of body color will be there. So wherever A is coming, that will come. Yes. Pleiotropic effect. Whether it will be a pleiotropic effect? No, this is not a pleiotropic effect. Okay. So that will not come. Yes. Wherever D is coming, it will not come. Then these genes are linked together. These two genes are independent assortment. What will be the right answer? These genes are not an independent assortment. Okay, they are linked together. Okay, so A and C will be the right answer. So from genetic, sure they are asking questions. Okay, from Mendelian inheritance, they are sure asking 1 or 2 questions. You have to read that one. Okay, that will cover both general agri portions also. Okay, which of the following database do not store nucleic acid data? They are saying this will not store nucleic acid data. Swissprot is particular for protein. This we have seen. This is a secondary database. This is a secondary database. Yes. So what will be the answer? Swissprot is a protein. Okay. For protein only we will use. Remaining gene bank, EMBL, DDBG, DNA database for DNA data bank for Japan, EMBL gene bank. This all for nucleotides. Clear? Or nucleic acids. Okay. Next, hormones that are to be used in tissue culture can be sterilized by. This you have to answer. Hormones that are to be used in tissue culture can be sterilized by which method? We can autoclave or flame sterilization, HEPA filtration or filter sterilization. We can use, yes, filter sterilization. Very good. Filter sterilization we can use. Expression of anti-nutritional factor or negative regulator can be eliminated by which of the following tool? They are asking just a genome editing tool. Which tool we can use to particularly regulate this anti-nutritional factor accumulation. Okay. See here, overexpression of encoding gene. This will pakka it will not come. Correct now. If you overexpress means that particular component will be accumulate more only. So wherever C is coming, that option will not come. Clear, simple way. RNA interference, yes. Gene editing technique, genome editing we can use, yes. The particular gene can be knocked out. Tilling, target induced laser lesion in the genome. This definition, this expansion they have asked one time. Okay. Target induced laser lesion in genome. Okay, not gene. In genome, they have given in the option gene or genome. Okay, so this is very important point. So A, B, D and E is correct. A, B, D and E is correct. Yes, very good. Which of the following is correct for the statement genetic code? They are repeatedly asking one or two questions from this genetic code. Okay, genetic code. It uses RNA ribonucleic bases to make a code. Yes, this will be there in mRNA. Okay, anticodon will be there. Okay, if you see UGS the start codon here, ribonucleotide bases only it's using. Yes, this is correct. Deoxyribonucleotide, it will never use the deoxyribonucleotide. So wherever option B is coming, that will be wrong. Okay, simple way. Wherever B is coming, that will be wrong. A single coding dictionary is used for almost all virus bacteria. Yes, this is also true. During translation, the codons are read one after the other with no breaks between them until a stop signal is found. That will also be correct. Yes, option A will be the right option. Given below two statement, they are asking about okay. A partially diploid bacterial cell for the transduced gene can be produced. Partially diploid bacterial cell. The partial diploid case is resulted due to complete transcription phenomena. The same question they have asked in 2023 shift 1. Okay. Just by changing the sentence. Okay. Generalized transduction means what like that they have asked. These both are correct only. Yes, option A is the right option. Yes. Okay. This is another important thing you have to read. Okay. Isolation of, they have given different technique. Isolation of CDS, plasmid, mRNA and proteins they have given, and they have asked to match this. Okay. Among them, if you see what genomic library is there, cDNA library is there. Okay. CDS sequence means that is what? That are coding sequence. Correct. Coding sequence. So coding sequence will always come in cDNA library. So 5 will come. Yes. Isolation of plasmid, alkaline lysis we will use to remove that plasmid outside. Yes. Isolation of mRNA. mRNA 3' will have AAA. So we can add oligo TT column. So here we will come to suppression of proteins. Gel electrophoresis we can use for both suppression of DNA and RNA molecules as well as protein. So this will come 1. 5, 3, 2, 1. 5, 3, 2, 1. Where it's coming? 5, 3, 2, 1. 5, 3, 2, 1. Here mistake is there. Okay, 2, 1. Okay, this is the right option. Okay. Next, amplification of known DNA sequence. Amplification of cDNA sequence. Selection of transformed cell. Calcium chloride. Okay, they have given different terminologies. We have to know, we have to match with the particular instrument we are using. Okay. So amplification of known DNA sequence. So known DNA sequence means we can easily design the primers. Okay, primers we can design, then we can amplify. So 3 will come. Yes. Only here is coming 3. Yes, very good, excellent. Option C is the right option. Amplification of cDNA sequence. There RT-PCR we can use. Selection of transformed cell. Colony PCR we can use. Calcium chloride. That will be specifically for what? Preparing competent cells. Yes, very good, right option only. Yes. Expressed sequence time from Marcus. Again one more question they have asked. Expressed sequence tags are short sequence obtained by sequence of cDNA clones. Yes, that is correct only. We can obtain, yes. EST can be used as STS. Whether this is correct? EST, express sequence tags, can be used as sequence tag sites. Yes, we can use that one. Okay. Plant hormone question, another important thing and simple question only. Which of the following hormone is preferred for rooting in plant tissue culture? For rooting, what we will prefer? We will prefer auxin or cytokinin, or what we will prefer for rooting? Rooting means high concentration of auxin should be there. Among these two are cytokinin. So the right option will be auxin A only. Yes, very good. Where is Indian Institute of Agricultural Biotechnology is located? Again, one more question from institute. So you have to be very much thorough about institutes. Very much thorough about institute, where it is located. Okay. So the right answer is what? Ranchi, Jharkhand. Okay. Ranchi, Jharkhand. Okay. Where, what is the correct extent form of GEC? This I have told in previous lecture itself. GEAC, Genetic Engineering Appraisal Committee. Okay. Genetic Engineering Appraisal Committee, not approval. Okay. Appraisal Committee. Apart from that, we have also, apart from appraisal, not approved. Okay. Not approved, appraisal. Okay. Then most important thing I have already told you, have to know definition about RDAC, expansion, recombinant DNA advisory committee, recombinant DNA advisory committee. Okay. Advisory committee. Then in last question, what they ask means IBSC, institutional biosafety committee. Okay. These are some of the expansions they can ask. Okay. So you have to be thorough about that things also. Okay. Next, centrioles. This we know, centrioles will be present only in animal cells. This is direct question, simple question also. Okay. So animal cells. In VK Jain, first unit itself they have given about the plant cell organelles. Okay. Cell organelles in the differentiation table, one table will be there. There they have given what are the specific organelles present in plant, animal, and what are the organelles that are common to both eukaryotics and prokaryotics. Okay, you just go through that one. VK Jain. Okay, fundamentals of VK Jain books have this one. Okay, next, PCR based DNA amplification is an essential feature of which of the following combination of molecular markers? They are using PCR. They are using for these markers. What are the markers that are based on PCRs? Okay, you have to see here. RFLP, restriction fragment length permission. There we will use restriction enzymes only. So this will not come. Here also RFLP is there. That will not come. Here also RFLP is there. It will also not come. So the right option is C. Here RAPD, random amplification of polymorphic DNA. Here we will use single primers. Okay. This we will say as 10 mers. 10 mer primer. Okay. 10 bases will be there. That's why we are saying 10 mer primers. Simple sequence repeats and amplify fragment length polymerism. Here restriction enzyme also we will use. Then PCR also we will use. That's why amplify fragment length polymerism is there. Okay. So the right option is option C. This is repeated question. 2024 also they have asked the same question. Okay. Same match without changing any option. Okay. This we already discussed. Orthologous means what? Homologous genes found in different species. So this will be the right answer. Okay. Simple. Splicing means removal of intron. Mass spectroscopy for protein fingerprinting we will use. Uniport for protein database. This we have already discussed. Okay. Next, the properties of RAPD markers. This we already saw. Okay. RAPD marker. It is what? It's a dominant marker. RFLP is a codominant marker. This is the first discovered marker. This is a codominant marker. Okay. This will have the ability to differentiate the homozygous and heterozygous. Okay. So RAPD is a dominant marker. So where is coming? It's a dominant marker. This is correct. Okay. Here single primer only we will use. Yes, this is also correct. Low specific and target band. No, this will not come. But low annealing temperature we will use. Yes. Yes, very good, very good. A, B and D. A, B and D. B is the right option. Students have already answered. Last question of today's session. Okay. Can we solve? What is the year of establishment of NCBI? TK. NCBI when they have established. TK. You have exam on July 4. Yes, July 4. JRF is there. TK. This NCBI was established in November 4. TK. November 4. 1988. Okay, this is important. November 4, 1988. Okay, November 4, 1988. With this, we are completing the first session. Okay, at 2:30, we will continue the remaining 60 questions. Okay, if any doubt is there, means kindly post in the chat box. Okay, any doubts are there? Any doubts are there? If no other doubts, means we can see in the next class. Okay, students. Thank you. No, sir. Yes. Okay. Okay. Thank you.